Good evening, everybody. My name is Jeff Prescott, and I'd like to welcome you to this evening's program, CAR T-cell therapy and high-risk non-Hodgkin lymphoma. So tonight's program, we're hoping for this to be an interactive presentation. We have some wonderful faculty that will be joining us, and I'll introduce them in just a moment. But tonight we're going to be talking about CAR T-cell therapy in a few different areas: relapsing, remitting DLBCL, indolent lymphomas, multiple myeloma, and some of the future directions. During the course of the program, we will accept questions from the audience, and we'll be able to share that with the faculty as we get a little bit closer and depending on how much time we have. You can be able to ask those questions if you look towards the bottom of your control panel. You can be able to hit the Q&A button and submit that to us. So with that, I'm going to introduce tonight's chair, Dr. Andre Gua. He's a physician-in-chief from Hackensack Meridian Health Oncology Care Transformation Service and chairman and chief physician officer from the John Thoreau Cancer Center. Dr. Gua?
Good evening, everyone, and welcome to this webinar that is going to focus on the exciting field of CAR T-cells. Tonight, without further ado, actually, I'm very excited to welcome our panel. We have Dr. Tatiana Feldman, who attended John Thoreau Cancer Center, in charge of the T-cell and Hodgkin lymphoma program. We have Dr. Lori Leslie, who is the director of the Indolent Lymphoma and CLL at John Thoreau Cancer Center. And finally, we have Dr. Noah Biron, who is a brilliant myeloma attending, to share her perspective as well on myeloma. So without further ado, this is a very exciting time. And I have to say that in this very difficult time of post-COVID recovery, we were very fortunate to have been able to keep our CAR T program open and have enrolled quite a few patients. And the reason being is that this is really a modality that allows us to give an option to patients who really have no other option. So without further ado, I want to introduce Dr. Feldman, who's going to talk about CAR T-cell therapy rationale and activity in relapsed refractory diffuse large cell lymphoma. Tatiana?
Hi. Hello, everyone. Thank you, Andre, for a nice introduction. I'll try to be brief because I'm sure everybody knows this type of information, that even though DLBCL is highly curable, about 30–40% of the patient will relapse. And on average, probably 50% will be salvaged with autologous stem cell transplant, but we still have 50% who will do very poorly and have terrible outcome with no efficacious means of cure or even prolongation of the life. The data which I would like to refer to is coming from SCHOLAR-1 and earlier on CARL data. SCHOLAR-1 was retrospective analysis, which looked into the patient who relapsed within first 12 months of a frontline therapy. An overall response rate to whatever chemotherapy was given, only 26%, with median overall survival is near 6.2 months. Follicular lymphoma, it's a low-grade lymphoma. We know it has excellent prognosis, and majority of the patients probably live normal lifespans. However, again, 20% of the patients who relapse within first 30 months of treatment do quite poorly. So, and here comes CART. With much ado, I would like to introduce the fairly novel, but already not so novel, new intervention, which basically trying to harness our immune system to kill lymphomas. So CART is chimeric antigen receptor T cells, and as opposed to all our other interventions which were tried unsuccessfully, such as vaccines, dendritic cell vaccines, immunomodulation, which really doesn't work that well in DLBCL, the CART is basically thing in itself. It has this chimeric antigen receptor, which is self-activating because of this either CD28 or 4-1BB domains, which are co-stimulatory domains. And it specifically targets in this particular, in lymphomas, antigen called CD19, but you really can come up with any target, any antigen, and create CAR T cells against anything you can find a good target. So I have difficulties moving it. So there are several generations of CARs. So first, second, and third generation, the ones which were approved. It's a second generation when a CAR stimulatory domain either for 1BB or CD28. And again, the advantage of CAR T that, number one, it's a one-time deal. It's given once, and this is living therapy. The cells will continue to do the work. It's really independent of HLA antigen recognition, so it's really universal application. It can target anything, and it can be active both CD4, CD8, and nowadays it produced pretty quickly from anywhere 3 to, on average, 3 weeks is the time of production. And there is obviously no risk of GvHD like allogeneic stem cell transplant, as these, most of them are autologous cells. So there are multiple CARs being developed, and this list is probably 1/5 full. There are 2 products which are currently approved by FDA. It kind of moves on its own. I'm not pressing anything. Okay. So one is called Axicell, or Trinam Yescarta. The pivotal trial based on which was approved is called ZUMA-1, and it's approved for adult patient relapsed refractory large B-cell lymphoma with prior 2 lines of therapy. And it could be any DLBCL, primary mediastinal, transformed follicular. And the second product is Kymriah. It's the same CAR, it's the same idea of the product, CAR-19. Pivotal trial was Juliet, and approval for 2 indications. One is adult with relapsed refractory DLBCL, and second, there is also approval for ALL. So this is a brief comparison of major trials which were reported within the last 3 years. The first 2 trials I mentioned, ZUMA-1, Juliet, 2 products already approved, and the last one, Transcend, was just presented at latest ASH by Dr. Abramson, which is Lysacel. That's another product which is also CAR-19. ZUMA-1 utilizes CD28 as a costimulatory domain, and Juliet and Transcend product utilizes for 1BB. So I just put some characteristics of the patients, which basically they were all diffuse large B-cell lymphoma, transformed follicular lymphoma, primary mediastinal, and Transcend also allowed grade 3B follicular. They were all relapsed. The number of patients over 100 in all 3 studies. As you can see, ZUMA-1 did not allow double hit lymphoma, but Juliet and Transcend had about 27 and 22% of double hit lymphoma. This is particularly aggressive disease. Patients were heavily pretreated, more than 3 lines of therapy in 69 to 50%. Although, as a lymphoma doctor, I'll make a comment, then maybe people who were able to survive more than 3 lines of therapy and 4 lines of therapy, they do have not such a bad lymphoma, because as we all know, DLBCL patients run out of time very quickly. So primarily refractory and refractory to last therapy, very high number, over 50%. And a good number of patients had prior to the stem cell transplantation. So obviously, ZUMA-1 had the longest follow-up, presented in Lancet Oncology, 27 months. So they presented 2-year data. Juliet is a little bit shorter, so 19 months. And Transcend is the latecomer, so the median follow-up is about 11 months. I couldn't correct this slide. Is also dose level 3, because Transcend presented phase 1, phase 2 data, and the dose level was 1.5, 10 to the 8th. However, bulk of patients, about 140 patients, received dose level 2. And as you can see, the data is very similar and quite interestingly very similar and robust. So best overall response was 82% in ZUMA-1, 73 in Transcend, and 52 in Juliet. However, the complete response is quite remarkable, and we don't see it in this disease. 54, 40%, and 53%. And what's interesting, I don't have the curves, but patient who achieve complete response by 6 months appears to maintain this complete response in case of Zuma, by 2 years. So this is the data of PFS at 12 months. As you can see, it's not really changing. 12-month PFS and 18-month PFS is very similar, and the overall survival is the same way. Transcend was reported a little bit differently. Median PFS was about 7 months. Overall survival is same, almost like 20 months. Patient who achieve complete responses, the median duration of response for all of the trial have not been reached, as well as progression-free survival of CR patient have not been reached. So this is across the different trial, different products. The results are quite phenomenal in patients who really had no options at this time. And we're seeing cure. Obviously, all the trial needs to mature a little bit more, particularly Transcend. But we are seeing the first cure of this patient population. So obviously, those patients were very heavily chosen. Patient had to wait in line to get on a trial. So the question, particularly aggressive lymphoma, may not have been, they were not excluded, but they were precluded de facto. So there is some real world data which was published by Nastupil is from MD Anderson and Jacobsen from Dana-Farber. So this basically were patients who were not able to get on the Zuma trial, but they were given commercial product. And what's very interesting that I'm pointing your attention to the efficacy, overall response 81% and 71%, with complete responses having 57 and 44. MD Anderson always gets better results. I don't know how they managed to do that. With median progression-free survival, very much similar to Zuma 1 trial with the similar 6-month overall survival. So with that, I would like to give the flag to the next speaker. This is a brief introduction of novel and very exciting therapy which is changing the outcomes of aggressive diffuse large B-cell lymphoma.
Dr. Leslie, if you can share your perspective on indolent lymphoma and CLL as well. Thank you. Thank you, Tatiana. That was great. We'll have some questions at the end. Thank you.
Yeah, thank you for introducing us to the field by going through DLBCL, Tatiana, and thank you, Dr. Guo, for the introduction as well. So today I will be reviewing mantle cell, marginal zone follicular, CLL. So we will go through this also briefly. So the first study we'll talk about is ZUMA-2, looking at CD19 CAR-T and mantle cell lymphoma. This was first presented ASH 2019 by Dr. Wong and published recently April of 2020 in New England Journal. We did participate and treat a number of patients on this study at Hackensack, and Dr. Hua was the PI at our site. So this is a multi-center phase 2 study. 74 patients were enrolled with relapsed or refractory mantle cell lymphoma. They could have had up to 5 prior lines of treatment, and everyone had to be exposed to BTK, progressing either on or after exposure to BTK inhibitor therapy. Primary endpoint, overall response rate. And looking at table 1 here, this is a very highly pretreated, high-risk group, in my opinion, with 31% of patients highlighted at the top having blastoid or pleomorphic characteristics. And also 88% of patients were refractory to BTK inhibitor therapy. That was defined as either primary refractory in 62% of patients or relapsed after initial response to BTK inhibitor therapy in 26% of patients. The primary efficacy analysis included 60 patients that had a minimum of 7 months follow-up. And the overall response rate in that group, shown in the panel here was 93%, with 67% of patients achieving a complete remission. As with other CAR-T studies, there was a portion of patients that initially had stable disease or progression of stable or partial response that later converted to CR, with a median time to conversion of just over 2 months. In terms of at a median follow-up of 12.5 months, 57% of patients on this duration of response curve were still in remission. And of those that achieved a complete remission, 78% maintained that remission. Looking at the PFS and overall survival curves also at the bottom at 12 months, 61% of patients. 12 months, the PFS was 61% and overall survival was 83%. Including the entire intention to treat population, which was the 74 patients enrolled, the overall response was 85%, with 59% of patients achieving CR. And looking at these curves here, depending on where you're looking, somewhere maybe as early as 9, 10 months, maybe as late as 12 to 15 months, it seems that the duration of response and PFS curves are flattening with a plateau at the end. Of course, the end of the curve has a very few number of patients at risk, less than 10, so becomes less reliable. But looking earlier, it does certainly seem to flatten, making us hopeful that for a portion of patients, this may be a durable response. In terms of toxicity, majority of patients had grade 3 or higher AE, mostly was cytopenias, 32% of patients had infections. In terms of the AEs of interest, which is cytokine release, 91% overall, 15% grade 3. There were no grade 5 events of CRS. Median onset similar to what we see in DLBCL, earlier median onset of 2 days with a duration of 11 days. In terms of neurotoxicity, 63% overall with 31% grade 3 or higher, and again, no fatal events, with a median onset similar to what we see in diffuse large B-cell lymphoma, slightly later at 7 days, with a median of 12 days to resolution. Some patients did have ongoing neurologic symptoms at time of data cutoff, mostly mild at grade 1 tremor in 3 patients, grade 2 concentration disturbance, and dysesthesia in grade 1 in 1 patient. There were 16 deaths on study, 14 were due to progression of disease. There were 2 grade 5 AEs, 1 due to organizing pneumonia, 1 due to bacteremia, and both those were attributable to the lymphodepletion chemotherapy. I think we'll hear a little bit more later about mechanisms of resistance, but of the 14 patients that did relapse, 13 had detectable CD19 at relapse, and of the 7 patients that had samples for evaluation, 5 of those 7 did have detectable CAR T cells, though detectable at a level that was below the peak expansion for that patient. There were a number of correlative studies done for ZUMA-2, similar to other CAR T studies. So the median time to peak CAR T was 15 days. And notably, CAR T expansion was associated with objective response, as well as achieving MRD undetectable status at the 4-week time point. It was also associated with toxicity, with a higher rate of grade 3 or higher CRS and neurologic events in patients who had higher peak expansion. So looking further into these correlative studies, there was another abstract presented by Dr. Wong at ASCO this year, looking at pharmacologic profile and outcomes of KTX19 in high versus low risk patients with mantle cell lymphoma and ZUMA-2. And this showed that the KTX19 cell had cell product had a slight bias towards CD8 cells, as well as an effector memory phenotype with a median CD4 to CD8 ratio of 0.4. In terms of the 29 patients who had MRD data at the 4-week time point, 83% did have undetectable MRD, and these patients had increased median cytokine levels with a peak of about 7 days, as well as median peak granzyme B and soluble PD-L1 levels. This was also associated with grade 4 neurologic events, but no other, in terms of the cytokine peaks, but no other adverse events were associated with higher cytokine peaks in this study. And there was no difference among low versus high risk mantle cell lymphoma types, which goes along well with what was previously reported with increased expansion associated with overall response as well as toxicity in patients on ZUMA-2. Of note, there's further studies going on with the same product, ZUMA-18. We are also participating in Hackensack. And that's looking again, phase 2, relapsed refractory mantle cell lymphoma, similar population, slightly more real world, and that bridging is allowed. So eagerly looking for further follow-up from ZUMA-2 as well as the follow-up from ZUMA-18. So with that, we'll move on to CAR T-cell in follicular and marginal zone lymphoma, so the indolent lymphomas. This is ZUMA-5 that was also just presented a month or so ago at ASCO. looking at CD19 CAR T cells in patients with relapsed or refractory indolent lymphoma. A phase 2 study planned 160 patients for enrollment, about 125 with follicular lymphoma and marginal zone lymphoma of 35. This is the interim analysis, including 80 evaluable patients with follicular lymphoma that had at least 9 months of follow-up. Also 16 patients with marginal zone lymphoma with at least 1 month of follow-up are in the interim analysis for efficacy. Key eligibility, relapse refractory, follicular, grade 3B is excluded, or any type of, or nodal or extranodal marginal zone lymphoma, at least 2 prior lines of therapy, including CD20 and alkylating agent, standard conditioning and cell dose, and the primary endpoint overall response rate. For the safety analysis, 140 patients are included. So this is the primary efficacy showing in the overall cohort an overall response rate of 93%, with 80% of patients achieving a complete response. Looking at follicular lymphoma specifically, overall response rate 95%, 81% of patients achieving CR, and in marginal zone lymphoma, 81%, 75% achieving CR, but of course smaller numbers with only 16 patients. Again, there was a portion of patients that initially had a partial response and later converted to CR. And the responses were consistent across follicular lymphoma subgroups in terms of risk stratification. In terms of duration of response, you can see the top curves here are follicular lymphoma and the bottom are marginal zone lymphoma. Numbers are quite small, particularly looking at the number at risk here for marginal zone lymphoma, so it's hard to extrapolate much from the curves other than saying overall survival, all patients were alive at the time of this data cutoff, which was a median follow-up of 15.3 months. So at that median follow-up of 15.3 months, where all patients had at least 9 months follow-up in the follicular lymphoma subgroup, the median duration of response was 20.8 months, with 68% of follicular lymphoma patients achieving an ongoing response. And of those in a complete remission, 80% of patients had an ongoing complete response. Of those in partial remission, only 18% of patients had an ongoing response. In terms of toxicity, again, this included the entire 140 patients. Almost all patients experienced at least 1 AE. The most common grade 3 or higher AE were cytopenias, neutropenia, anemia. Grade 2 or there were 2 grade 5 AEs. One was multisystem organ failure due to CRS, that of course was attributable to the CAR T cells. The second was aortic dissection that was thought to be unrelated to the process. Here are the data for the incidence of CRS and neurologic events. Any grade for CRS, 79. A low grade of grade 3 or higher, only 8%, with the common symptoms being common fever and hypotension. Median onset was slightly later than we see with diffuse large B-cell lymphoma and mantle cell lymphoma at 4 days with a duration of 6 days. In terms of neurologic events, the time course was a little more standard for what we're familiar with in other types of B-cell malignancies, with a median onset of 7 days and duration of 14 days. And again, any grade 58%, grade 3 lower at 17%. Development in CLL has been a little more challenging for a variety of reasons compared to the other B-cell neoplasms. So first, the treatment landscape has been really rapidly evolving since the approval of ibrutinib in 2014. And mostly these patients are treated with a string of targeted therapies compared to other B-cell malignancies that use a lot of chemoimmunotherapy more standardly. There's also been some, during development, issues with CAR T expansion, and it's been difficult to sort out why that is. There's some emerging data that a combination with ibrutinib for a variety of reasons, either as the most recent line of therapy or in combination with CAR T, may be an appealing approach to overcome some of these barriers we've had with CAR T expansion. This could be because ibrutinib or BTK inhibitors help mobilize the CLL cells out of the lymph node environment. There's been some evidence that ibrutinib specifically can improve T cell number and function, and also there's an association with enhanced CAR T proliferation and anti-tumor efficacy preclinically in combination with ibrutinib. So this is just some data from a study presented or published in Blood this year by the Fred Hutchinson group with 19 patients treated with CAR T cells and concurrent ibrutinib starting 2 weeks prior to infusion and at least 3 months after. High overall response rate of 83 with 61% achieving MRD undetectable status, and a lower rate of CRS, with the same peak expansion that had been seen in other patients with CLL. So this is very promising, and there's a number of studies ongoing, some of which we have at Hackensack, including ZUMA-8 and Transcend CLL, both looking at CAR T-cell, CD19 CAR T-cell therapy in CLL with or without ibrutinib. So just a brief mention of some of the longer-term toxicities. So CAR T-cell therapy is obviously a complex treatment that's only done at certified treatment centers. So there's some things that a referring doc may need to think about when the patient's being referred back to their initial referring center. So we've talked a lot about the short-term toxicities. I won't go into that, but in terms of longer-term, things like cytopenias can occur, though mostly this happens during and immediately after CAR T-cell therapy. There is an incidence of more prolonged cytopenias, and usual growth factor support, transfusion support can be used to manage those patients. Also, obviously, work up for any underlying causes, including secondary malignancies that can happen post-CAR T. Many of these patients are heavily pretreated and could be associated with any of their prior therapies. And then also hypogammaglobulinemia is something that frequently may need to be evaluated in our patients who are post-CAR T once they go back to their referring physician, similarly to as patients would be monitored and managed post-high-dose chemotherapy or a stem cell transplant. And I think that is it for me. So with that, I will pass it on to Dr.
Go ahead. Go ahead, Laurie. Go ahead, please. Go ahead. I'm sorry.
I said I'm done there. So unless there's any questions, I'll pass it over to Dr. Biran.
We have a few questions, and then we'll keep them, try to give them for the end. Dr. Biran, please, on myeloma. That was great, Laurie. A lot of data, and we are very fortunate at the Adjunct Cancer Center. We have been involved in this cell therapy approach now since close to 10 years, and with the NCI initially, and have treated a lot of patients commercially, but on clinical trials, and have been at the forefront of all this. And I think this is thanks to our research team, CAR T-cell team, cell therapy team, Dr. Donato's team, and Kate Bertrin particularly, to organize all these cell therapy setup at every time that is kind of customized. So we really appreciate that. Without further ado, Dr. Biran, myeloma.
Thank you very much. So the CAR T-cell world in myeloma is different from that of non-Hodgkin's lymphoma in that we don't have any CAR T-cell approved yet, but certainly we are very close, and we are learning from our lymphoma colleagues on how to manage a lot of these cytokine release toxicities and how to manage short and long-term toxicities. Myeloma has been a rapidly emerging field in terms of therapeutics over the last decade. Here you can see some of the newer treatments that have been approved. Most recently this year, we've had a number of therapies approved. Despite the advances in myeloma, we still have a very poor prognosis for patients who are refractory to CD38 antibody therapy. And you can see in this study retrospectively done on 275 patients from 14 academic centers, patients who are penta-refractory, so refractory to 2 PIs, 2 IMiDs, and a CD38 monoclonal antibody, median survival is 5.5 months. So certainly there's an unmet need for the development of new therapies. And BCMA has emerged as a novel target in myeloma. It's an antigen that's expressed on plasma cells as well as healthy plasma cells as well as the malignant lymphoma cells. And it's a TNF superfamily receptor that has a key role in B-cell maturation and differentiation. It promotes myeloma cell growth and chemotherapy resistance. And we see that there's increased BCMA expression with progression from an MGUS to advanced myeloma. And BCMA is really the most common antigen that's being used in CAR T-cell therapy. There are other therapies that target BCMA that are non-CAR T-cells, such as the bispecific T-cell engagers, as well as antibody drug conjugates. So BB2121 is one of the most advanced CAR T-cell therapies in development. It's a second-generation BCMA-targeted CAR T-cell. So it has a signaling domain made up of 4-1BB signaling and a promoter region, as well as the anti-BCMA binding domain. And a lentiviral vector is used to produce the CAR T. And you can see this data was published. This is the phase 1 data that showed CRB401 study. Median time to partial response or better was 1 month. And with doses of greater than 150 times 10 to the 6 CAR T cell, median PFS was 11.8 months. And that has become the therapeutic dose now used in almost all of the trials. In the dose escalation part with doses less than 150 times 10 to the 6, the median PFS was much shorter. And see, in patients who achieve MRD negativity, and that's within the first 2 months, the median PFS was 17.7 months. So early achievement of MRD negativity correlates with a longer duration of remission. This is the KARMA study for patients with relapsed refractory multiple myeloma, multi-center single-arm phase 2 study. We are participating in this study here at HAC. Patients with advanced disease, 3 or more prior lines of therapy were enrolled. Patients are leukapheresed and then can receive bridging therapy. And then they receive lymphodepletion with Cytoxan, fludarabine, followed by their CAR T cell infusion. And now we are up to 450 10 to the 6 CAR T cells. 54 patients were enrolled in that cohort. Primary endpoint is overall response. And some of the key secondary endpoints are achievement of CR and, of course, safety. So baseline characteristics of these patients, very heavily pretreated patients, median prior lines, 6, and ranging 3 to 16 prior lines. 35% were high-risk cytogenetics, and that tends to be higher than what we see in most trials. Usually we see 20, 25%. And you can see 94% of these patients were refractory to anti-CD38 monoclonal. And you can see that the overall response rate, and we'll focus on doses greater than 150, were 82% in the 450 times 10 to the 6 dose, with a CR rate of 39%. And that's a median follow-up of 14 months. And you can see that expansion of CAR T cell and persistence of CAR T cells correlated with response. And that's what this study shows. Median time to peak expansion is 11 days. And that helps us in terms of when to expect cytokine release syndrome, which correlates with median time to peak CAR T cell expansion. And this is the KARMA study. Here you have event-free at 12 months, 78% were PFS at 12 months. And OS is not yet mature, but you can see the median overall survival, 19.4 months. And this is by, so you have the higher doses are what are achieving higher median progression-free survival and a longer duration of. Next, we'll go on to the CARTITUDE study, which is the LEGEND J&J BCMA targeted CAR T cell therapy. It also uses a 4-1BB costimulatory domain. 2 different epitopes that bind simultaneously to the BCMA antigen. The first study was done in China. That was the LEGEND-2 single-arm phase 1 trial. And patients were 3 or more prior lines treated with increasing doses. That study achieved a median PFS of close to 20 months with CR rates of 28.2 months and median overall survival of 36.1 months. The more mature study is the CARTITUDE-1 trial, which is the phase 1b/2 trial. And we'll go into that one. So the response rate for the CARTITUDE study was at 100% overall response. and 97% VGPR or better, 86% stringent PR or better, with a 9-month PFS of 86%, and that's close to a year median follow-up. And you can see the median time to first response was very short. So very, very promising results so far with all of the prior to CAR T. The third one that we'll discuss is the EVOLVE trial, which is the Juno autoleucel for relapsed refractory multiple myeloma study. And you can see that the results are comparable. This study also used Cy and Flu for lymphodepletion, and overall response rates in the higher dose cohorts range between 89 and 95. And this table compares characteristics across the 3 cell trials. You can see age was similar, high-risk cytogenetic, similar. The differences here, median prior lines also similar. The EVOLVE study had a slightly higher risk of rate of triple refractory, and ranging 63 to 90% required bridging therapy. Safety, you can see also very comparable across all of these trials. We see decreased ANC in the majority of patients, 90 to 100%, decreased platelet count, and CRS all and grade 3 or more, 84%, 89, and 93, but grade 3 or more, not very high, 3 to 7%, grade 3 or higher CRS. So slightly lower than we see in our lymphoma patients. Median time to CRS, 1 median up until 7. So the CARTITUDE CAR T cells have a higher time to peak expansion, and that correlates with a longer time to median CRS. And then here we have infections. Depending on the follow-up, we see anywhere between 40 to 70% for all grade. About 50 to 80% required tocilizumab, much less percentage required steroid use, 15 to 52%. Anakinra was used in about 0 to 25%. And efficacy, remember, these are very heavily pretreated refractory patients. So to see an overall response rate ranging between 73 and 100% is very impressive in this patient population with stringent CR, 33 to 86%. And the reason the KarMMa has lower response rate is probably because they're including lower dose levels in this study. Median PFS not reached, and 8.8 months. And so where are we heading? I would say BCMA is definitely a promising target. The question is going to be, how do we increase the duration of response? How do we achieve persistence of CAR T cells? These current clinical studies so far are demonstrating high response rates in a heavily pretreated population with acceptable toxicity. But we need to further understand mechanisms of resistance and relapse. Maybe we need to use CAR T cells earlier in the lines of therapy, so that's being evaluated. And what are the next generation approaches? How will we improve duration of response? And that's the key question.
Thank you. That was a very, very exciting on myeloma. We'll have a few questions. And without further ado, I will actually go briefly on some of the perspective on next steps. So this is really a revolutionary therapy. This became the first live therapy, gene-modified therapy that was approved. It provides durable remission, as you see, in patients who have really failed all the standard therapy and have no option, and potentially cure, particularly in patients with a relapsed refractory, heavily pretreated lymphoma, diffuse large cell. You saw that 37% of the patient can do really well, up to 40% over 2 years, and very few late relapse, mantle cell early, but very impressive, follicular as well. Axi-cell and T-cell, we heard, are both approved, and in aggressive lymphoma, and T-cell in ALL under 25 years old. There's a lot of issues that are remaining, and we started to hear some of it, is that the relapse rate is still high. And on the right, you see the salvage after CAR T failure in aggressive lymphoma is very dismal. The only really data is somewhat lenalidomide checkpoint, particularly BiTE antibodies. But these patients have a poor outcome because they're not great candidates for an allo transplant in that setting. Patients with CNS involvement with lymphoma, that is becoming more an issue, obviously, in that setting. Toxicity, we heard about CRS and the ICANN. Immune cell therapy is associated neurotoxicity. And what is the best timing in the combination? And also, what is the resistance mechanism and how we can overcome them? So we heard about toxicity, and a part of the next step, we have learned the toxicity, how to manage much better by mostly using anti-IL-6, either receptor for TOSI or IL-6 cytokine with cetuximab. This is given earlier, more preemptively, and it does not impact the T-cell amplification and the outcome. And IL-6 is one of the cytokines responsible of this CRS syndrome. Steroids have really had an impact, particularly in neurotoxicity, and there was a data that showing that you can also start to do this more preemptively without affecting the durability and the responses. There's other things that Noah touched upon. Anakinra is a suppressor of myeloid, anti-IL-1, can suppress myeloid cells. That is also a good source of IL-6. There's early data, and there's ongoing phase 2 in severe CRS and neurotoxicity. JAK inhibitors, ibrutinib. Ibrutinib is an interesting story. It decreases the CRS and actually has been used, BTK inhibitors, with some interesting data also in COVID in the situation of this hyperinflammation. BTK can be protective from the COVID, as we discussed, but it can improve the efficacy of T cells. And in this patient, there's actually a rationale that Laurie mentioned briefly, rationale to use BTK combination with CAR T cells, and pre-CAR T cells, pre-collection, harvest, but also afterwards, and improve the outcome. And finally, there's, for the toxicity, there's the off-switch factor that is very important, either introduce a suicide gene to control the proliferation, or built-in receptor that you could turn off, for example, with rituximab, and we'll mention that in a minute. So how do we improve the efficacy and focus on large cell lymphoma here? We know that the prognostic factors, that this is a heavily pretreated population, and the standard prognostic factors that we look at, bulky, age, IPI, etc., or the tumor cell of origin, really doesn't have an impact. The CD4-CD8 ratio really doesn't have a big impact either. But what we know is that what's really critical is the peak of the CAR and the CAR under area under the curve. And it really correlates with the response and durability. The CAR T persistence is something that has been really questioned, how important it is. And it turns out that there's obviously, again, different beliefs in the system. The 4-1BB may be potentially more durable, CD28 higher amplification early on, but in a ZUMA-1, about 1/3 of our patients in long-term remission didn't have any detectable CAR T cells. And Laurie alluded to hypogammaglobulinemia that actually recovers over time as this patient can recover some B cells. And we heard about TOSI and steroid use as well. That doesn't affect the outcome. So the mechanisms of resistance are tricky, and then we're not going to go into the detail. They're not fully elucidated, but they serve as a rationale, as we'll see, to try to develop the next generation of treatment and CAR T cell therapy. So there's no clear diagnostic test yet to inform a management decision when we do CAR T cells in our centers. We don't have any good test to evaluate the CAR T cell epitope expression at the surface of tumor cells or the CAR expression on T cells. We look at the number, obviously the viability of the T cells and the number of T cells that we generate, but it is very important to look at understanding, deep dive a little bit into the mechanism of resistance that are very complex, and I just will highlight a few. One of them is obviously access to tumor site, and this is very important because the trafficking is something that is not fully elucidated on how we manipulate this, but it's tricky in solid tumors where there's additional physical barriers with extensive stroma-rich extracellular matrix that can prevent the access to expand in solid tumors. In CNS tumors, glioblastoma, there's some interesting data with local injection of CAR T. And we know that CD19 CAR T cells can penetrate the blood-brain barrier and potentially, actually some data in primary or CNS, secondary CNS lymphoma mostly, where the results has been somewhat impressive and this is being tested more. But we don't really know yet when we inject systemically, when someone has CNS lymphoma, how much cells we need there to control that. And there's some interesting early data, but looking at built-in chemokine receptor to change this trafficking and improve the diffusion of those CAR T cells. And there's an anti-CD30 in Hodgkin and CTCL that is also expressing CCR4 that potentially facilitate the diffusion of those and of those CAR T cells. The second mechanism of resistance to CAR T is really the CAR T, the factor of the CAR T expansion that, as I mentioned, correlates with the clinical response across the board. The CAR T persistence, as I mentioned, is required in ALL. In myeloma, it's not entirely clear yet, but it's not required in large cell lymphoma, as I mentioned, because we have seen that some patients have no CAR T detectable and still in remission. The lymphodepletion to get a favorable amplification of those T cells matters. That's why we do Cy/Flu and not Cy alone. There's some interesting concept looking at retreatment, small experience, usually not as durable, but also splitting the dose, and potentially that might be helpful. We know that some of these CAR T constructs can be recognized and lead to an immunization, and therefore we can reinfuse them. So there's concept of humanized construct that are entering the clinic. And finally, the checkpoint inhibitors combination. We all have had this patient that had CAR T cell progression. We give them checkpoint inhibitor. We have seen some expansion of the CAR T cells and some additional response. So this was tested in the ZUMA-6 trial in combination with anti-PD-L1 and showing the longer expansion and higher expansion of the CAR T cells. And finally, the next generation, third and fourth generation, is to either do these armored CAR T cell with cytokine to reinforce the immune amplification locally, or also to have more stimulatory domain to continue to expand on those CAR T cells. Another mechanism that is a resistance is the loss of antigen expression. It's a clonal selection process. It's either a splicing variant, so you lose the epitope, particularly in lymphoma, the lineage switch where you lose the CD19 expression fully more frequently in ALL, and there's something called trogocytosis, which is actually rare but interesting, the transfer of the CAR T receptor to an effector cell, natural killer or CTL, and that will obviously induce the loss of the effector cell. So how do we address the loss of antigen expression? And I'll allude in a minute about dual CAR T cells, and there's many ways to go into these CAR T. It's a dual receptor, it's two CAR T at the same time, or it's two different receptors at the surface to try to connect with two different antigens to try to prevent the clonal selection. And this is being taken advantage of in a number of clinical trials, CD19, CD20, CD19, 22, I will see in a minute. Increasing the target at the surface of the tumor cell seems also very appealing. And there's some data from a gamma secretase inhibitor that increases BCMA at the surface of myeloma cells with some early data, but are promising. Finally, the real key is a mechanism of resistance that is either intrinsic to the T cells themselves or intrinsic factors, mostly immunosuppressive. So the intrinsic factors, there's the concept of T cell fitness, and we heard from my colleagues tonight that probably treating some of these patients earlier matters because their T cells are not so beat up with multiple lines of therapy and evolving of underlying malignancy, which is by itself immunosuppressive. The concept of CAR T cell exhaustion is a very important. It is due to the tonic signaling by the CAR T construct itself. So how do we address this? Well, we have next generation CAR T design. We also have tests that are not ready for the clinic, but this will happen, that polyfunctional index that will measure the T cell fitness. There are some studies that are looking at having a balanced CD4-CD8 ratio, and then and that's an interesting approach where there's potentially better efficacy. It is important to have no leukemia and lymphoma cells during the manufacturing process. So, for example, in Kitex 19 in mantle cell, the T cells are purified and removing the tumor cells. And this is really important because otherwise the potentially contaminating tumor cells could overstimulate and exhaust the T cells during the manufacturing process. And I think one of the best factors is the allogenic CAR T cells, because those are healthy T cells and potentially will be very helpful in that setting. The intrinsic factor is more the exhaustion from the tumor burden, the immunological milieu that is poorly characterized yet. There are some gene expression profiling and signatures that are starting to look both, as I mentioned, at T cell fitness, but also looking at the immunological environment. And the microenvironment, the checkpoint blockade is something very important. We see an overexpression of PD-L1 in some of these tumor cells that exhaust the T cells. So how can we address this? Is combining with checkpoint inhibitors, and also in the future looking at gene editing to make those T cells immune from suppressive signaling. So very quickly, some example of these T cells. So I mentioned taking advantage of the dual CAR T, and here this is a combination, the Alexander study presented at ASCO. I thought it was interesting, dual CAR T and checkpoint inhibitors. It's a phase 1/2 study of what's called AUTO3, and it's CD19, CD22, and bicistronic chimeric antigen so dual receptor, and then that in diffuse large cell lymphoma relapsed refractory, in combination with checkpoint inhibitor. And this is really important because the results is very interesting. So this is a dose escalation process. The responses were interesting, 75% response rate, 63% CR at the dose of 150 million and with a day -1 and pembrolizumab. As you can see here, this is not a very long follow-up, but you can see some of these responses are very prolonged. 10 of the 11 CR are still ongoing. There was 0 grade 3 CRS toxicity, and there was 1 grade 3 neurotoxicity. And the recommended phase 2 is at 150 to 450, 10 to the 6, with pembro on day -1. The second study that I wanted to mention is the Alpha study. This is a first example of allogenic CAR T cells, the in large cell lymphoma study from MD Anderson and other centers, presented by Dr. Nilapu at ASCO. And this was the first in human data. So the concept here is that you take an allogenic healthy T cells, and by using the CRISPR technology and TALEN technology, the gene editing, you can remove actually the T cell receptor, and you also remove CD52 here, and you're going to use those cells that are allogenic. And the advantage is that you can treat all eligible patients. You potentially could repeat dosing. There's no need for complex logistics. This would be faster off the shelf and less issues with product variability made from healthy T cells and issues with T cell fitness because these cells have never seen chemotherapy. And the way this trial was given, so they gave the 501, which is a cell therapy, and the 647 is a CAMPAT equivalent to further immunosuppress the host and the Treg. And then you can see here, there was no GVH, there was no DLT, only 1 grade 3 CRS, and no neurotoxicity. The response rate was very impressive with 63% CR response rate and 37% CR. 75% of the responding patients remain in response at the data cutoff. It is not a very long follow-up, but as a proof of concept, having a less toxic, easier to use, off-the-shelf anti-CD19 concept is very appealing. So in summary, we have a very impressive pipeline of CAR T cell. This is the first live therapy approved. The toxicity is more and more manageable, as I mentioned. The focus is really, as we heard from all our colleagues, increasing the CR durability. And one of the factors that I'm really convinced, and we have ongoing studies, particularly in the second line in large cell lymphoma, 3 studies open at our center. 1 is actually completed, 2 of them are enrolling, and looking at second-line therapy versus stem cell transplantation. And the reason to do this is that we know that patients with an early failure in large cell lymphoma do poorly with radiotherapy and transplant. So these studies are ongoing. It is important to refer these patients very early. The other thing is we need biomarkers, and we'll have this during the discussion, but there's no clear biomarkers to predict the outcome. But what is really interesting is that there was some data presented at ASH a year ago, and looking at cell-free DNA at day 28. And the patient who have cell-free DNA negative at day 28 did remarkably well. This was not a large population, but it makes a lot of sense. But it also offers an opportunity maybe to do combination when we monitor this patient. Remember, when we heard from our colleagues tonight, we have a significant 20 to 30% of patients who early on just have a PR or even stable disease. And sometimes it takes a year to go into complete response. And I think this is very important that we don't forget this is a live therapy and that we can continue to potentially modulate it. The next generation CAR-T in combination with checkpoint immunes and small molecules, as I mentioned, BTK, and molecules fighting toxicity. And finally, NK CAR-T cells that are very early, but allogenic from cord blood, for example, and with a very high yield, you can amplify 7,000 times the NK cell you generate from a cord blood, and very low toxicity. And we don't know about the durability yet, but it seems to be also a potentially interesting source of cell therapy. So this is just a graph. We're not going into the detail that shows you on the top the different therapy types that are being looked at, the different target in the middle, and on the left, this is the rapid increase of the number of clinical trials that are ongoing, thousands of trials that are ongoing in cell therapy. So I think this is a really, really exciting time, and I think that we need to not forget to refer patients for this, because this is really something that is a game changer, and probably even when you do this, better. So thank you, and then I want to switch to some questions that we have. And the first one I want to ask Dr. Feldman is that when you see a patient with a CAR-T coming for CAR-T cells, what makes you feel like this patient is going to actually respond well and have a durable response in that setting? If you can comment. Predictive markers, basically.
So basically, I don't think that, ZUMA-1 has the longest follow-up, and I don't think there was any particular patient characteristics which would make you predict which patient will respond better, which patient will respond worse. What we all know, and these patients actually never made it to the trial, but a few patients which were, if someone has a rapidly growing disease and rapidly progressing, these people probably will not do well with CAR-19 unless some successful bridging therapy will be utilized, which, as you know, ZUMA-1 did not allow bridging. So the information which I'm making my opinion is based on real world data where they did use bridging therapy. However, very interestingly, people who needed bridging therapy, they tend to do worse, which probably goes back to the parameter of the disease. You have sick patient who needs to get chemo before getting CAR, as opposed to someone who has slowly growing disease, so they don't need any bridging therapy. What we do know that double hit lymphomas appear to do well. So that's the biggest concern. Do those cells work on double hit lymphoma? The answer is yes. And the second question, what about tumor bulk? Do people with a high tumor bulk respond to CAR-19 therapy? And remarkably, CAR-19 can kill any amount of disease, and that appears to be not a negative factor. So those questions have could be in a consideration of not sending the patient. So bottom line, there were some characteristics, as what Andrew was alluding to, the low persistence of the cells, etc. But it's still part of the research, and that's why the products are being developed to increase the persistence and expansion of the cells. But from the patient standpoint, I don't think there were any particular characteristics.
Thank you. Dr. Leslie, can you comment on the impact? Follicular data was also very impressive. It's not a very long follow-up, but particularly in a population that is high risk, and where do you see this is going to fit? And did the prior therapy impact the response to those patients?
Yes, I think this is very exciting for the fields of follicular lymphoma. Patients in the study had a median of 3 prior treatments, about a quarter had prior auto transplant, which honestly I feel like is probably underutilized in the current treatment landscape of follicular lymphoma. About a third had PI3 kinase inhibitors. everyone had CD20 immunotherapy and alkylating agents. So, patients at that time point would be kind of considered for clinical trial or maybe even allogeneic stem cell transplant. So looking at CAR T as an option for that patient population, I think is much easier to tolerate overall compared to something like an allogeneic stem cell transplant, and the responses are very encouraging. The follow-up, I agree, hasn't been long enough to really see that nice plateau that we're hoping for, as we're starting to see in mantle cell and see in diffuse large B-cell lymphoma. The challenging thing about follicular lymphoma and indolent lymphomas in general is we don't have any good predictive or prognostic markers in the frontline setting to identify those patients who will have more aggressive courses from the beginning. But as with other types of lymphoma, I think the future of CAR T in follicular is first, hopefully, approval in the relapse refractory setting, and then moving it earlier in the treatment landscape for those high-risk patients, like progression of disease within 24 months of starting their initial therapy. Whether, extrapolating from CLL, we'll start using MRD more and maybe trying to consolidate patients who are still MRD detectable at the end of whatever induction therapy. That's where I think the future is. At the same time, I think indolent lymphomas have a little bit of a challenge when it comes to potentially toxic therapies like CAR T. Because generally you think, oh, these patients will do well for a long time, even though, of course, that's not always the situation. There's a different acceptance earlier in the treatment landscape for potentially serious toxicities, including an episode of grade 5 CRS in ZUMA-5 than potentially there would be when we were doing ZUMA-1. And most of those patients were otherwise potentially candidates for hospice. So it's a different kind of acceptance of potential toxicity. So I think compared with DLBCL and the indolent lymphomas, that's kind of another balance that's going to be a little more challenging as we advance the CAR T field.
Well, what was interesting in the original study that from the NCI, among the first few patients that had lymphoma treated, a few of them were follicular lymphoma, and they are well over 5 years, and they have done really well. And it seems that this has been potentially more durable. Time will tell. And Dr. Biron, can you share with us a little bit in myeloma? The data is across the board very impressive by the brisk response and reaching MRD negativity very quickly. And we still see a significant amount of relapse. How do we lose the control of the disease when those CAR T cells are able to drill down and bring a patient with lots of disease, heavily pretreated, refractory, and bring into an MRD negative status and relapse relatively quickly?
We don't fully understand the mechanism of relapse yet in these patients. We know that there is some signal that loss of CAR T cell persistence correlates with relapse. We do see an increase in extramedullary disease after relapse from CAR T cells. So we don't know if there's loss of BCMA antigen. It's hard to tell. Is it just that we're dealing with a very heavily pretreated population who's going to have severe extramedullary relapses no matter what treatment they were given, or is it a reflection of the CAR T cell therapy, making the disease more aggressive and resistant? We really have not fully those concepts as far as I know.
Where do you see more and more you're able to treat these patients further and further, but you also now can identify very early on very high risk patients for standard therapy. How do you see them shifting towards more CAR T earlier on?
There are ongoing studies looking at CAR T cell in first remission. So sometimes even after collection, stem cell harvest for auto transplant, but not yet proceeding with the transplant. So doing it before transplant or as consolidative therapy. There's also off the shelf CAR T cell allogeneic products that are being evaluated in high risk patients earlier in the lines of therapy. So first line in these ultra high risk. There are a subset of patients who we know are not going to have a survival of more than 2 years in multiple myeloma. So in that subset, we are, as Dr. Leslie said, accepting toxicity earlier on in the lines of therapy.
Thank you. Dr. Leslie, you reviewed the mantle cell lymphoma data, and this is, for our audience, a patient who have failed chemoimmunotherapy and BTK inhibitors, usually the outcome is not great, to say the least. And I think that those curves are still early, but I mean, it was very impressive. How do you see them in this therapy in the future fitting in mantle cell lymphoma, knowing that we have high-risk patients that do very poorly with standard therapy?
I think mantle cell lymphoma is probably the most challenging subtype of lymphoma, just because it has the characteristics of an indolent lymphoma with the tendency to relapse and also that baseline chemo refractoriness. I think, at least in my career, CAR-T and mantle cell is probably the biggest development, and I think is really going to revolutionize the treatment of relapsed refractory mantle cell lymphoma. I'm encouraged about the interaction with BTK inhibitors in the subtype of mantle cell, and I think moving forward, maybe that's how we'll improve efficacy and decrease toxicity, allowing us to move earlier in lines of therapy. We're already doing kind of a risk stratified approach for patients with very high-risk disease, TP53 abnormality at baseline, and I think using CAR-T again to eradicate MRD detectable patients or otherwise consolidate high-risk patients earlier in their lines of therapy is going to hopefully be something we're doing in the near future.
Yeah, I think as a team, and all of us, we have treated a lot of mantle cell lymphoma, lymphoma and CAR-T patients. And then it seems clearly that the take-home message would be, I think that as these therapies become more and more available, there will be additional approval beside large cell lymphoma soon. To refer these patients and to offer them this treatment before they receive too many therapies, I think this is going to be a key factor in the durability. One last question for Dr. Feldman, maybe. Where do you see CAR-T, let's say in lymphoma now, have a role as a consolidation?
So it's a very good question. There was a study done at Memorial. Craig Sorter reported it over a year ago, where they were using, the protocol was high-risk DLBCL. First relapse, patient would get, they would have to get salvage chemotherapy and have a little bit of a disease present, so they couldn't be CRs, and then would get autologous stem cell transplant as standard of care, followed by a CAR-19 infusion, very much at similar time as stem cell infusion. And the data, again, granted it was phase 2 study, there was no direct comparison, but at least historical comparison, I don't think it showed much of a difference. And the question which I would have is that you do need the presence of an antigen, not just benign B cell, but for the expansion. And that's my question, to which I don't have an answer, whether complete response and no evidence of the disease is actually a negative factor for CAR therapies. However, I think this issue needs to be explored more, particularly with now MRD analysis available and high-risk disease states, whether it's mantle cell lymphoma or still imagination diffuse large B-cell lymphoma, and also whether we can minimize upfront therapy from dose-intense regimens to less so, if you can use consolidation. But I think it's something which is being explored, and the studies actually are ongoing. And there is also a study ongoing at MD Anderson, which utilizes CAR-19 in a frontline therapy. But I don't think we have answers to that.
Thank you very much. Well, now it's time it went by very fast. There's so much to cover in CAR-T, in B-cell malignancies, and there will be studies, reopening studies in other diseases, including solid tumors. So I would definitely I want to thank our audience and thank our faculty for a great presentation. This is a remarkable time to be in oncology, and we have more and more options to offer our patients. So I hope that we can remember about cell therapy, and I think this is going to change the way we look at therapy in cancer in general. This is part of immunotherapy. Was at a talk a while ago when someone had a talk called cells, not pills in the future, and I think this is the beginning. Without further ado, thank again for the audience, and thanks to my colleagues and the faculty, and thank you guys for organizing this. Have a great evening. Good night.
Thank you. Bye-bye.
Bye-bye.
Thank you, Dr. Gua. Thank you for our faculty.